Hello, everyone, and good and welcome. So today we're going to be covering a part one of a long two-part class where we dive into plant form, plant function, plant structure, and then really sort of take a deep dive into what are the sort of the nutrients plants needs, what does the environment that plants grow in look like, and all that fun stuff. So we'll start off with plant form. It's sort of the most basic aspect of plants. So plants, very much like humans, have a overall body organization. Right? We think about sort of how our body looks like. We have tissues and organs and blood vessels, and there are many analogous structures in our plants that we'll discuss. Now, a plant that is vascular in nature consists of a root system. This anchors the plant and serves to absorb water and ions as well as other nutrients from the soil. And then we have the shoot system, which is basically all the above ground mass that you see on a plant, including stems, leaves, flowers, bark, so on and so forth. So anything above ground is what we typically classify as a shoot system. And there are repetitive units that we'll discuss, including node, internode, leaf, and axillary buds. So this is our sort of standard plant diagram. This is a flowering plant. We have the apex of the shoot. We have some flowers, and we have some other pieces here that we'll dive into in just a moment. But we have the shoot and the roots, and there's some anatomy and some overall structures that we'll discuss here. Now, plant cell types vary based upon a few key factors. So first being the size of the vacuoles. So you'll remember from Bio 1, the vacuole is, sort of analogous to like a digestive or a holding tank for the plants. They hold lots of water in their vacuoles, and they can also digest things inside their vacuoles as well. Now, we can also classify these plants whether or not they're living or at maturity. And there are some aspects of plant biology where the cells are actually just completely non-functional. You could essentially consider them dead. Then we can also think about the thickness of secretions found in their cellulose walls. So some have only a primary cell wall, others have more heavily reinforced cell walls with multiple layers of cellulose. So we'll get into these, but there are three basic tissue systems we need to think about when we're discussing our plants. The first being the dermal tissue, which is the outer protective cover, the ground tissue, which function in storage, photosynthesis, and secretion. And then finally, we have vascular system, which you can make analogous to the blood system, which conducts fluids and dissolves substances throughout the plant's body. And then we have basically we sort of think of as human tissues, right? But we have tissue systems where each of these types extends through the root and shoot systems within inside of our plants. So the cell wall, as we've mentioned, is sort of an important distinguishing factor here. Now, we can have sort of a standard cell look right here with the large vacuole, the nucleus, and all the other organelles that we, know and love. And they have that cellular membrane very much the way we do. But they have this first primary cell wall, and you'll notice that we have the cell membrane, sort of that inner layer, very much like we have. And then you have cellulose fibers that run parallel and that anchor and sort of essentially go through insides of the cell, anchoring into the microtubule system. And so you'll notice that cellulose runs in parallel fibers. So if you've ever say looked at the husk of a corn, you kind of pull that apart, the husk of a corn apart, it really looks like there's fibers in there. That's what you're really getting at, right? Those are cellulose fibers running horizontally along the outer membrane of these plants. So that's the primary cell wall. And as we mentioned, plants can essentially thicken their cell walls by adding more and more layers, as we can see here. So we have, over time, in this plant on the right, we have primary cell wall, which has been pushed out by the secondary and primary, I'm sorry, to the two secondary cell walls here, one and two. So plants can have differing levels of cell walls being very thick or being very thin, but at the end of the day, they all occur because of the presence of cellulose as a really important carbohydrate in this system. So now, new growth of plants grows at what we call meristems. Now, these are clumps of small cells that have very dense cytoplasm and very large nuclei. And they basically function as stem cells, right? So you think about what stem cells are in animals. They're, you can find them in the umbilical cord. Technically, your bone marrow is also stem cells because it differentiates into different blood types. But the idea with the stem cell is it can differentiate itself into any tissue type. Now, on the human side, we think of like differentiation into an organ or skin or your brain, right? But in plants, it's differentiating into different types of tissue, right? Root, shoot, guard cells, and so on and so forth, as we'll see. Now, these cells are essentially the active sites of cellular division or mitosis in our plants, and they act as the extension mechanism for roots and shoots by what we call apical meristems. So meristems is where the new growth occurs, but apical meristems basically push the plant out into the world. And then the other aspect we have here, what we call lateral meristems, they produce increase shoot and root diameter. So they push essentially they make the plant more girth, have more girth. So, and we can look at what's going on here. We have a meristem cell. Again, thinking of it like a stem cell. It divides, producing another meristem cell and then some sort of differentiated cell. But that meristem cell that was then produced can then go and divide, producing another meristem cell and another differentiated cell, and so on and so forth. So they always produce one meristem cell and one differentiated cell. So they can always sort of keep up this act over and over again. Now, the topic we're going to focus on for this moment is what we call apical meristems. These are at the tips of roots and shoots, and they give rise to primary tissues, which we are collectively call the primary plant body. Now, apical meristems themselves are composed of cells that need protection. In particular, we have them protected by root caps in our root systems and leaf primordia in our shoot systems. So let's look at what we got here. So we have shoot apical meristems here. So we have older leaf primordium here. We have the shoot apical meristem. We have what we call a lateral bud primordium. And then we have young leaf primordium here. And this has been stained nicely on this left-hand side where it's the same side of it's the two different sides of the same plant. But we can see we have dermal tissue, we have ground tissue here in this sort of light color. And then we have vascular tissue here in this sort of darker color. But again, you'll see that the outer leaf primordium essentially protects the new budgeoning, essentially shoot apical meristems, protecting them from the outside world. Because you can imagine if you're a relatively young plant, you want to be protected from the environment because you don't want the environment to kill you when you're very young, right? And then we switch gears looking at the roots. Now we have the root apical meristem. Here, now we can see the vascular tissue, the ground tissue, and then the dermal tissue. But then we have this root cap right here, this overall sort of, you can think about like almost like a pen cap, right? Where it fits over, protecting the root apical meristem as it grows. The root caps, harder cells, they protect the root meristems from the overall environment or surrounding them in the soil. So that's what we have in terms of meristems. They're protected, but again, they are the sites of cellular division and growth and girthening of our plants. Now, this leads us to different tissue systems. Now, these apical meristems give rise to three distinct tissue systems. We've already mentioned a few of these, but we'll just go through them again. There are three primary meristems. First is the protoderm, which forms the epidermis, which you could think about like your epidermis, i.e. your skin. Then we have the procambrium, which produces primary vascular tissue, and then the ground meristem, which differentiates into ground tissue. So again, forming the three distinct tissue types we see in our plants. Now, some plants also have some what we call intercalary meristems, which arise in the stem internode and add to internode length as well. So let's focus on lateral meristems first. So they are found in plants that exhibit what we call secondary growth. Primary growth is vertically, secondary growth is horizontally, right? So, as you grow from a kid to an adult, right, you're growing vertically, right, which is your primary growth. But if you get, say, chunky, you're getting wider, thus exhibiting secondary growth. So it's kind of a easy analogy to think about there. Now, lateral meristems give rise to secondary tissues, which are then collectively called the secondary plant body. Now, typically speaking, this growth is actually what we see in woody plants. So, most trees that you see out there are woody plants. So we have a couple pieces. We have the cork cambrium, which produces outer bark, the vascular cambrium, which produces secondary vascular tissue, and secondary xylem being the main component of wood. So let's actually just physically look at these apical and lateral meristems. So we have the primary stem, secondary stem, secondary roots here at the top, and then we have the primary root at the bottom. And you'll notice they have relatively similar structures to them, right? They have their meristems, they have some sort of protective tissue, but they have distinct sort of an anatomy. But again, there is a high level of similarity between the two of them. But we'll see the most homology here between the primary stem and the root stem. Very, very similar in terms of structure and function because they're doing the same thing, right? Whereas the secondary ones are growing wider, thus they have very similar structures and functions as well. Now, there are three distinct types of plant tissues, as we've mentioned. First is the dermal, which is on the external surfaces that produces some sort of protective covering. We have the ground tissue, which forms several different internal tissue types and can also participate in photosynthesis, store things, and provide structural support. And then vascular tissues, which conducts water and nutrients. Now, the vascular tissue are going to be like your vascular tissues, thinking about movement of things around the plant body. Um, Next up, we have the dermal tissue. So sorry, let me take a step back. We're going to dive first into the dermal tissue, which forms, as we mentioned, the epidermis. It's one cell layer thick in most plants, but it can be slightly thicker in others. Now, it does form the outer protective covering of the plant, and it is covered with a waxy cutin layer consisting of what we call the cuticle. And it's not cuticle as in like, what you see on your nails or whatever. It's just like a nice waxy outer layer. So let's talk about some structures on the outer epidermal layer. The first is the guard cells, and these are, as you'll notice, they look like sausage-shaped cells, or they look like really, really plump lips, as you'll see in a moment. But these flank the stoma. The plural for this is stomata, which are epidermal openings, which allow the passageway for oxygen, carbon dioxide, and water. So let's actually just look at these cells. So we have the epidermal cells here and surrounding these. But here we have the stoma, and when we have multiple ones as we see down here or over here, we have stomata. It's kind of counterintuitive to me. I always thought stomata should be the singular and stoma should be the plural, but that's just not the way it is. Now you can see the stoma, again, it's just an opening. Now the reason you have an opening, remember there's a waxy cuticle layer on the outside of this. So as you guys probably know, waxy cuticle layers don't allow readily for the movement of gases or liquids. So the stomata allow for the ready diffusion of gas and liquids through this waxy layer inside the plant cell because plants really need gases, both CO2 and oxygen, to be exchanged with the environment for them to be plants, right? Because that's what plants do. But again, you can see the guard cells here. Again, they look like sausages, or maybe they look like really, really plump lips. You can think of it however you want. I prefer the sausage analogy, but they do kind of look like mouths now that I started looking at it. We can actually look at a kind of an interesting mutant in plants. So we have the too many mouths stomatal mutant. Now this is actually in a plant called Arabidopsis thaliana. It's just a mustard plant. Now, this mutant actually lacks essential signaling pathways for the spacing of stomata, so it actually causes this really weird patterning. But stoma are usually sort of paced and separated by sort of a distinct amount, as you can see here, right? They're always separated typically by one epidermal cell. There's this overall layer of structure, even though you might not think it there is, but there is. And there are mutants out there that create faulty structures of of stoma on the underside of these plant leaves. Now, I will just make one last mention of this. These stoma are only found on the underside of a plant leaf. They're not found on the upper side of the plant leaf. Why is that? Well, stoma act to help regulate water loss. And if you're on the upper side of the leaf, it's really hard to regulate water loss if you're always being cooked in the sun, right? Because that's what the top side of leaves always face. Now, next is in terms of another structure we see on plant epidermal tissue is what we call trichomes. Now, these are cellular or multicellular like hair outgrowths on the epidermis. They occur frequently on stems, leaves, as well as on reproductive organs. Now, they help reduce evaporation by covering stomatal openings. They also protect leaves from high intensity light and UV light, and also help with temperature fluctuations as well. Now, there are some trichomes that actually are what we call glandular, so they're just glands. You can think like your sweat glands that actually secrete substances that help inhibit herbivory. But we can actually physically see glandular trichomes here. We have the trichome, which is this entire structure. Now, normally these are just going to be like they'll essentially look like a lollipop, which just have one sort of bulb at the end of the trichome. But in this case, they're glandular, so they have four little sort of components to them, and again, they secrete a level of sticky substances that helps help trap insects and protect the plant from herbivory. So in this case, these are actually on tomato plants, which helps them protect them against herbivory from different types of insects. Now, trichome patterning is actually controlled by a gene. And so I'm not going to dive too deep into this, but just to know that trichome patterning is controlled by a couple different proteins, and you can mess with trichome patterning in plants by turning these genes on and off. So that's the sort of the epidermal tissue of our leaves. Now let's dive into the roots, where we'll talk about the epidermal tissue of the roots, where we could look at first at root hairs, which are essentially tubular extensions of individual epidermal cells. Now, the primary function of roots is to absorb water, ions, and other nutrients from the soil. Now, the best way to do that is to have the most surface area. So our roots act as essentially a mechanism to increase surface area in our roots, thus increasing the efficiency and efficacy of absorption. Now, these just want to make note of one thing. These are root hairs, not to be confused with lateral roots. Lateral roots, as we'll see, are just actual meaningful roots sticking out. They have more girth to them. But these are, again, like your own hairs. So that's our epidermal tissue in roots and shoots. Now we'll switch to talking about ground tissue, which there are three types, and I think they're kind of fun to say. We have the parenchyma, the collenchyma, and the sclerenchyma. And now let's dive into these three types, first leading with the parenchyma. Now, the parenchyma is by far the most common type of plant cell out there. It is what we call living protoplasts, i.e. they live for many, many years. So you can think of them like just like your average everyday tissue in your body. It lives for many, many years. Now, these typically function in storage, photosynthesis, as well as secretion, and only have primary cell walls. So their cell walls aren't too rich with cellulose as well. And they are significantly less specialized than the other types of plant cells that we'll see. Now we can look at some parenchyma cells here. Again, they only have the primary cell wall. In this case, it's a grass, but the cell walls are very thin, and you can see they're chock full of chloroplasts, which are all these little green bodies we see littered throughout this picture. And you can also see the nucleus here, just as an FYI. But again, lots and lots of involvement of photosynthesis, storage, and all that fun stuff. Now we have the collenchyma, which are flexible storage plant organs. Now they are really kind of cool. You can think of them almost like you know, like rubber. You know, they're very flexible but tough. They can bend without breaking, and they like our parenchyma are living cells, so they can live many, many years. But they also do lack those secondary cell walls. Now we can look at our collenchyma cells here. They do have a slightly thickened cellular wall, as you can see here in pink. This is from an elderberry tree, just as a note. Now, in these collenchyma cells, the thickened areas typically occur at the corners of cells where they meet other cells. But again, these are collenchyma cells involved in structural support around them. Which leads into our final cell type. These are sclerenchyma cells, which have very thick outer cell walls. Usually, they're typically speaking, they're not living at maturity, which is kind of a weird thing to think about, that, you know, potentially part of a plant's body is basically just not living. But in fact, many sclerenchyma cells are basically non-living, which one thing that commonly characterizes sclerenchyma cells in woody plants is actually secondary cell walls containing lignin. Now, lignin is one of the features of tree bark that gives it its really strong sort of toughness to it, making trees like, you know, like for instance, I have a wood desk right in front of me, and I can knock on that wood desk. It's very, very tough. Why is that? Well, that's because lignin. Now, in our sclerenchyma cells, we have two types. We have the fibers, which are long, slender cells grouped into strands. So, like if you think about like paper is made from tree pulp. Those are the fibers that you're thinking about. And then we have sclerids, which are variable in shape. They're typically branched, but they can occur in many or single groups. So let's physically look at this. So these are sclerids. Again, kind of a funky looking cell. I've always personally thought they kind of look like neurons, but they're not, they don't obviously don't function the same way as neurons, but they have that sort of similar shape to neuronal cells. Now, in this case, these are sclerids, which we actually can term as stone cells. And they are directly attached to living, sort of active tissue. And I don't have a picture of the fiber cells, but again, fiber cells are just long, fibrous tissue. Now we have vascular tissue. Now, again, vascular tissue we think about as like your blood vessels, right? Or your blood vessels or your arteries are just part of your vascular system, right? Moving water, moving ions, moving blood, oxygen throughout your body. And plants do the same thing. Right? We're not unique in this fashion, right? So the xylem, which conducts water and dissolved minerals throughout. Now, typically speaking, the xylem pulls things from the roots up to the shoots. And then we have the phloem, which conducts a solution of carbohydrates, commonly sucrose, but also other things such as hormones, amino acids, and other substances for plant growth, typically from the shoots down to the roots, but also through other parts of the body. So if you don't sort of remember these technical definitions, xylem goes up, bringing stuff from the roots to the shoots, and phloem goes down, bringing stuff from the shoots down to the roots. Now, there are sort of overall structures of xylem cells and phloem cells that we'll talk about. So the first for our xylem cells, we have what we call vessels. These are continuous tubes of dead, and again, kind of weird to think about, but it's dead cylindrical cells arranged end to end. Then we have tracheids, which are again dead cells that essentially taper at the end and overlap one another, um, forming this overall structure. So let's just take a peek at what we have here. We have our vessels here, and we have our tracheids here on the inside. So this is what electron micrograph is going to look like. Now you notice we have our vessels again pushing things up through our overall plant tissue. They have what's called a perforation plate. You can think of a perforation plate actually kind of like a filter, and again, because it kind of looks like a filter, and it actually acts to filter out not so good things from the liquids that are flowing through the vascular system, at least going up. And it can be a wide range of things, but we have our vessels and our tracheids both acting to take liquids from the roots up to the shoots. Why? Well, plants need nitrogen, plants need water, and where do you get that stuff from? You get it from the shoots. I'm sorry, you get it from the soil, right? And that's what we have. Now we have our phloem. Now phloem takes stuff from the top down to the bottom. Now, phloems have two types of elongated food-conducting cells. The first are the sieve cells, which we find, as we'll learn, in seedless vascular plants and gymnosperms, and sieve tube members, which we'll find in angiosperms. So we'll talk about what these groups are later in this lecture, but you only find them in certain types of plants. And now these are living cells, unlike our xylem, and they do contain clusters of pores, which we call sieve areas or sieve plates. So it's similar to what we just saw. And then we also have what we call sieve tube members, which are more specialized, which are associated with what we call companion cells, which should actually physically carry out many metabolic functions needing to maintain the sieve tube member. So let's look at this, and I like this picture because it really sort of highlights how similar, mostly because it's the color, right? But how similar this system is to our own vascular system, right? Our vascular system does the same thing. It's red. This vascular system is stained red. But again, we have water flow and nutrients moving down through this system. We have plasmodesma, which are those slight openings that allow the lateral flowing of water. We have cellular membranes here. We have these sieve tube members. Again, being part of this overall system. We have the nucleus and the companion cells, which again help maintain this sieve tube system. And then we have the sieve plate, which again functions in a level of support and as well as filtration. So that's the structure of our vascular tissue, xylem and phloem. Now, the roots have a similar pattern of organization and development, and I'm sorry, simpler pattern of organization and development than the stems. Now, there are four distinct regions in our roots: the caps and the boundaries. And the boundaries are divided into the cellular division zone, the elongation zone, and the maturation zone. So let's physically look at this in 3D structure. So we have the root cap down here. Again, that's that protective layer. We have the zone of cellular division. We have the zone of elongation and the zone of maturation. They all do slightly different things. So let's talk about each of those four areas. So first is the root cap. There's nothing quite like them in stems. Just as a note, root caps are unique in structure to the roots, but they contain two distinct types of cells that are formed essentially continuously by the root apical meristem. The first is the comella cells, which are the inner cells, and the root cap cells, which are the outer and lateral cells. Now, as we've mentioned, they function as protection because remember, these protect the meristems, which are delicate tissues evolved in essentially elongation and widening of these plants. And the other kind of fun thing root caps do is actually they're involved with the perception of gravity, which is kind of a weird thing. We think gravity is sort of this constant thing, but you have to be able to perceive it in some shape or form. And the the root caps help them do that. And just to sort of show you the root cap again, we have the apical meristem here, we have the comella cells, the root caps, and the outer cells here. So this is our root cap down at the bottom. Next, we have the zone of cellular division, which is essentially derived from a series of rapid divisions in the root apical meristem. Now, these contain mostly what we call cuboidal cells. They have very small vacuoles and a very large central nuclei, and they are the daughter cells of the apical meristem. So remember, the apical meristem is the site of active growth. They're essentially, stem cells, but again, that's where they're derived from in this zone of cellular division. Now, the apical meristem daughter cells will also subdivide into three primary tissues: the proderum, the protoderm, the procambrium, as well as the ground meristem. Now, there's actually really interesting control over this area from a genetic pattern. The first is the werewolf gene, and then the scarecrow gene. They do two distinct patternings. We're not going to cover these in much depth, but I figured I'd put them up there in case you're interested in genetics of this overall area. Now, this leads us to our second to last area, which we call the zone of elongation, where essentially this is where the root lengthens because the cells are basically becoming several times longer than they are wide. So you can basically think about like, I don't know if you ever had taffy before, but you think about like how you can grab taffy and pull it and stretch it. That's basically what's happening in the zone of elongation. Now, it's mostly lengthening the cells, but the width does increase some sense. Um, uh, and essentially, this is what we call the mature part of the root. And except for the increasing girth, they essentially remain stationary for the life of the plant, which is kind of an interesting thing. And then finally, we have the zone of maturation where the elongated cells essentially become differentiated into specific cell types. Now, the root surface cells become epidural cells. I'm sorry, epidermal cells, not epidural. It's the wrong word. Means completely different things. But the root cells, surface cells do become these epidermal cells, which have a very thin cuticle layer, include root hairs and non-hair cells. Now, we also have our parenchyma cells in this area, which are produced by cortex or the ground meristem. And there are inner boundary, which becomes the endodermis or the Casparian strips. And then we have the stellae, which are plant tissues that are interior to the epidermis and form what we call the pericycle. So we can actually physically just look at what this looks like. So we have the phloem and the xylem here, so our vascular tissue. We have the pericycle, we have the Casparian strip and endodermal cells, and then we have the cortex here. Again, all this connects together, pulling in water from the outer parts of the environment, nutrients, pulling it in, allowing it to get to the xylem to take it up to the shoots of the plant. And we could put this all together looking at a root cross section and physically see what we have. Now, there are two distinct types of root systems. We will discuss the monocots and the eudicots. You'll notice that the monocots are slightly more complex than the eudicots. We'll dive into like sort of the separation of this, taxonomically in a few slides. But again, we can physically see the structure here. Right? This is the cartoonized version, but again, we can see all the things that we see here. So we have our epidermal layer out here. We have the cortex. We have the endodermis, where the Casparian strip is. We have the phloem, the pericycle, the xylem, and then we have another inner part, which we call the pith. And we can see a similar thing here, but again, much, much simpler. The endodermis, which is the Casparian strip, is the xylem, the phloem, and then we have the outer epidermal layer here. So that's our roots. And just to sort of put this all together, we have this really handy flow chart. We have the stages of different plant tissues. You start with a fertilized egg. It differentiates either into roots or shoots, and then you develop all sorts of different pieces along the way, leading to bark, leading to wood, leading to roots, elongation, and so on and so forth. So again, a nice little handy chart that shows you sort of all the things that we have. Now, roots are pretty cool structures. We typically don't tend to think about roots unless we're eating the roots. So like, for instance, if we're eating potatoes, right, that's a part of the root system. So potatoes are pretty awesome because French fries and baked potatoes and all that fun stuff are awesome. But roots have some pretty interesting structure. Now, there are two distinct types of root systems, which we call the taproot system, which is a single large root with smaller branching roots. You can think of like a carrot, right? A carrot is just taproot. And then we have what we call the fibrous root system, which is sort of the typical root system we think about. Many smaller roots coming off one larger root that have relatively similar diameters. So two distinct root types, but some plants have some really interesting modified root systems with specialized function, which we call the adventitious roots that typically arise from any place other than the plant's root. So let's dive into some of these. These are some of the ones we're going to talk about. Not all of them, but we're going to talk about some of them overall. The first is what we call the prop root system, and now we see this in corn. You can see this is the main stem of corn, and the prop roots actually come out above ground of the corn plant and essentially embed themselves in the ground, acting as a tissue, helping to keep these plants upright. And for those of you that have never seen corn in person, these are really kind of funky. They really look alien-like in person. It's kind of a weird thing. But again, corn grows really, really big. These help keep corn upright. Next, we have aerial roots. So in this case, we have an epiphytic orchid, which attached to a tree. And aerial roots allow plants to attach to surfaces in the absence of roots. So specialized surfaces require specialized adaptations. Then we have the pneumatophores, which are actually my favorite type of root, which are found in plants such as mangroves, but basically in plants that grow in areas that are waterlogged, i.e., like coastal marshes. These plants will send out these pneumatophores that basically poke out from the soil, grow up out of the water and into the air, and allow them to uptake oxygen in these pneumatophore roots and bring it into the plant. Why? Well, because waterlogged soils are anaerobic, i.e., they have no oxygen, and plant roots need oxygen. Right? Everything needs oxygen. Well, not all bacteria, but every living plant needs oxygen. Then we have water storage roots. So in this case, we have an example from the pumpkin family. And these basically act as tissues that store large amounts of water. And so some of these plants in very, very dry areas can actually produce water storage roots that weigh well over 100 pounds or 50 kilograms. So pretty big roots, again, act as water storage. You can see them, they're very starchy, very similar to, like almost like potatoes. Then we have the butyrus roots, which are, in this case, it's a tropical fig tree. Now, these roots are essentially large roots above the ground, providing considerable stability for these very, very large plants. But these are butyrus roots. Um, Nothing special to them, just structure. So that's our roots. Let's dive back into our stems. Now, stems contain three types of plant tissue, as we discuss, and they also undergo growth from cellular division in their apical as well as the lateral meristems. And these shoot apical meristems initiate stem and tissue, intermediating producing what we call primordia, which develop into leaves, shoots, and flowers. So again, just to sort of show what we have the root apical meristem here, young leaf primordia, i.e. living tissue of the plant, and then we have the older leaf primordia, um, essentially, as we've discussed, protecting the young tissue. And we have whorled, like we see in sweet woodruff, where it basically is a 360 circle around a central point. Now, the external stem. The axillary bud, which develops into branches with leaves or many form flowers, and then the terminal bud, which extends the shoot system during the growing season. I know that was a lot, but luckily we have a nice handy dandy picture of a twig with all these things. So we have the terminal bud, an axillary bud, the node, the internode, the pellicle, the petiole, the blade, the terminal bud, and so on and so forth. So this is what we have. Again, this is the sort of overall words, but again, nice handy dandy picture for here to know the structure of our leaves. Now, inside the stems, we have a vascular system, as you would imagine. Now, as we discussed, there are two major groups of vascular systems, the monocots and the eudicots, and basically just reflects a differential level of organization. Now, monocot vascular systems, as we've seen previously, their bundles are usually scattered throughout the ground tissue system, whereas our eudicot vascular system is arranged in a ring with internal ground tissue, which we call pith, and external ground tissue, which we call the cortex. But again, here's our two distinct groups. So we have, I always get these, I always miss these some hits. So we have the eudicots here at the top and the monocots here at the bottom. Again, monocots very simple. Xylem, phloem, ground tissue, vascular bundle, epidermal layer on the outside. And again, the eudicots here at the top, much more structure, much more organization to them relative to the monocots. Now, vascular tissue arrangement is directly related to the stem's capacity for secondary growth. So in our eudicots, a vascular cambium develops between the primary xylem and phloem, which connects the ring of vascular bundles. And in monocots, there is no vascular cambium, so there is no secondary growth overall. So we can physically look at what we have here, our secondary growth. We have our primary xylem, primary xylem, the epidermis. Now, as this plant is exhibiting secondary growth, it's growing out, so it's pushing outward, extending out, producing secondary xylem and secondary phloem, which is ultimately, as we'll notice, going to be connected by the vascular cambium, so they're all connected in one shape or form. Again, getting larger and larger over time. But you'll notice that we sort of exhibit the secondary growth. It's all connected, but you can see these tree rings here. We have the annual growth ring. So again, it's pushing it out year by year by year, giving it that level of secondary growth, which leads us to talking about the tree stump rings. And you guys have probably heard of tree stump rings at some point in your life, but these are the rings in the stump of a tree which reveal the annual patterns of vascular cambium growth. The size of the tree rings depends on growth conditions. Um, and that's how we get our tree stump rings, again, from just the production of more and more vascular cambium, giving rise to these rooting systems. I'm sorry, these stumps. And we can physically see what's going on. We have xylem, which is comprised of the heartwood and the sapwood. Then we have the vascular cambium, which again is producing those tree rings. We have the phloem, the cork cambium, and the outer bark. And this is what our tree looks like. So if you ever wonder why a tree looks the way it does, it's simply because of the organization of differential tree tissue. Now, let's dive into some of these things that we mentioned. So the first is the periderm, which is comprised of the cork cambium, the cork, and the phyloderm, which forms the outer bark. And you can see we have the periderm here, which is the outer bark. And then we can see our parenchyma, which are those living sort of functioning cells along the inside of the tree. We also have a specialized part of bark, which we see in trees such as birch trees, which are called lenticels. Now, these are cork cambium that produces what we call suberized cells that permit gas exchange along the thing. So we have these essentially these cells here that are essentially holes. You sort of think of them as like pores almost. They allow gas exchange directly into the stem or the trunk of the tree, allowing for gas exchange directly. Now, there are a number of modified stems, bulbs, corms, rhizomes, runners, tubers, tendrils, and cladophylls. That we'll talk about a few of these to sort of show you some of the really interesting modified stems that we'll see. So the first is a modified stem called a bulb, where it has very fleshy leaves, a knob-like stem, and adventitious roots. This is very similar to like green onion. They produce these. Garlic produces a similar structure as well. Tulips, that's another great one. Then we have the rhizomes, which are here, these sort of overall. So we have the photosynthetic leaf here. We have rhizomes. These horizontal stems extend photosynthetic leaves upward and adventitious roots downward. Again, giving more anchoring points, more areas for photosynthesis, more areas for nutrient absorption. Then we have actually the Pachysandra plant here. I have these growing at my house. They're pretty cool. Now you'll see the normal plant here. It produces its standard root system here. But actually, one of the ways it does grow is it sends out these runners, where these are above ground roots. And you'll notice that along the runner, a new plant has popped up, and this plant will then anchor itself with roots and extend the runner out more and more and more. It's kind of a cool thing. Then we have stolons and tubers. Um, you can see the stolons are these little root bits here, and then we have the tubers here. Now this is a potato plant, and so obviously the best of all the plants out there, because potatoes are awesome. But this plant has underground stems with long internodes, which we call the stolons, and the tubers, which are swollen tips that help store carbohydrates. Now, the carbohydrates we're thinking about here is starch. So I'm sure you guys have probably heard that potatoes are a very starchy vegetable. Well, why are they a starchy vegetable? Well, the potato plant uses them to store products of photosynthesis, i.e. starch. We have tendrils. Now, these tendrils grow through the air and wrap around the stems of other plants for support and aid of climbing. We commonly see these on parasitic plants as well as vines. Then we have cladophylls, which are essentially what we see for cacti. Now, these are in this case, we have the stem of the cacti. It's flattened and it's photosynthetic, but the leaves have been modified with spines to essentially give them not only photosynthetic capacity, but also a level of protective capacity as well. So that's our cacti, cladophylls. Now, we sort of mentioned a little bit about leaves thus far when we talked about these cladophylls here, but let's dive more into the leaves, because clearly the leaves are an important part of the plants, right? They're the site of photosynthesis, right? They're the sort of the driving force behind the plant's biology. Now, leaves are initiated as primordia by the apical meristems themselves, and they are the site of photosynthesis. They expand by cell enlargement as well as cellular division, and they determine it in structure, and so their growth does stop at maturity. So they don't grow indefinitely. They reach a maturity and they stop growing. So there are two distinct morphological groups of leaves out there, whether or not you noticed them. The first is the microphyls, which have leaves with one vein that branches from the vascular cylinder of the stem and does not extend the full length of the leaf. This is found only in the phylum Lycophyta. We'll talk about the phylum Lycophyta later in this lecture. And then we have the megaphils, which have several to many veins, and this comprises most plants. So within within the sort of two groups, in particular the megaphils, we have our eudicot leaves, and we'll talk about the monocot leaves in just a moment. But most eudicots have leaves that have a flat petiole. Remember, the petiole is the part that attaches the stem to the leaf. Um, and now the leaf flattening increases photosynthetic surface, and then the flattening of the leaf also reflects a shift from radial symmetry, i.e., top to bottom symmetry as well. So we'll show you what that means when we talk about these two plants. But we can actually physically look at this, right? We have the shoot apical meristem, the top of a developing leaf, and the bottom of the developing leaf. Again, flat surfaces giving a level of symmetry here. So there are parts of plants, additional parts of leaves that we'll discuss. The first is stipules and veins. Now, leaves have these stipules, which are essentially the outgrowths at the base of the petiole, which may be leaf forming or potentially modified as spines. And then we have veins, which are vascular bundles within leaves. Now, typically the main vein in a leaf is parallel in most monocot leaves, and the veins of eudicot leaves often form sort of like an intricate network. So we have our monocot leaves down here, again, horizontally parallel veins, as you can see here. And then we have our eudicot leaves at the top, again, forming like this branch-like structure coming off the central main vascular leaf, right? And these are veins in the sense of vascular tissue, right? They're part of that vascular system, transporting nutrients around the plane. The plant, not the plane. Now, leaves can be simple or compound. Now, simple leaves contain individual blades. They might have teeth, indentations, or lobes, whereas compound leaves have blades that are divided into leaflets. So we have compound leaves here and simple leaves here. So nothing exciting, but again, two distinct types of leaves. Now, one of the more interesting parts of the leaf is their epidermal tissue. Now, the leaf's epidermal tissue is covered by a transparent epidermal cells that typically do not have chloroplasts, but the epidermis also has what we call that waxy cuticle. And we discussed the waxy cuticle, right? Thus far, it prevents loss of water, which is a really important thing for plants. But plants also have special types of glands as well as trichomes on their leaf's epidermal tissue. And as we've mentioned before, the lower epidermis, i.e. the bottom side of a leaf, contains numerous stomata flanked by guard cells, which we've previously looked at. But again, this is the structure of a guard cell. Sausage-like cells allow for the transport of gas through into the plant and out of the plant as well. Now, mesophyll is an important part of leaves as well. Now, most eudicot leaves have two types of mesophyll. The first, what we call the palisade mesophyll, which usually occurs in two rows of tightly packed chlorhynchoma cells. And then the spongy mesophyll, which is loosely arranged cells with many air spaces in between them, which help function in gas exchange as well as water vapor exit. And then monocot leaves typically have mesophyll that is usually not differentiated to either of these. So again, you can see the eudicots are simply more complex than the monocots, and that does reflect a overall level of evolutionary complexity as well, which we'll get to at the end of this lecture. So we have the upper epidermal layer here at the top. Then we have the palisade mesophyll, again, acting as that sort of bridge between the upper epidermis and the rest of the plant. Then we have the spongy mesophyll, again, very, very loosely packed, allowing for gas exchange. And you can see the different veins of the leaf here, and then you see the stomata here at the bottom on the underside of the epidermis. Again, air comes in here, goes into the spongy mesophyll, and then is distributed to the veins, which can go places, or it's distributed into the palisade mesophyll. And then any water vapor or any CO2 that is produced or oxygen can then be essentially passed through the guard cells, through the stomata, and out into the environment. Sorry, I had a sneeze. And again, this is what we're looking at, exactly what you're looking at, but I just a more 3D diagram of it. Again, these two are exactly the same. This is just a handy dandy 3D diagram if you do not like electron micrographs. I prefer this one, but again, this one's probably a bit easier to understand. Now, there are many different types of leaves out there, which we call modified leaves. So we can have floral leaves, which we call bracts. Now, these surround true flowers and behave as showy petals. We can have spines, which help reduce water loss and deter predators. Reproductive leaves, which are plantlets capable of growing independently into a full-size plant. And then we have window leaves, which are succulent cone-shaped leaves that allow photosynthesis underground. Yep, you heard that right. Photosynthesis underground. Kind of a cool thing. So let's talk about shade leaves first as another type of leaf, because I think it's a pretty cool leaf. So shade leaves are essentially very larger in surface area, but they have less mesophyll than sunlit leaves. Why is that? Well, if you're in the shade, you get less direct sunlight, thus you need more size, more mesophyll cells to increase your photosynthetic capacity. Then we have insectivorous leaves, which trap insects. Are plants. And that's a lot of stuff to remember, I know, but you're sort of looking for the big pictures here. What are the structures, the main structures? What are the overall, main functions of these tissues and so on and so forth? So again, a lot of stuff in those past 87 slides. But again, we're mostly focusing on the big picture here. Now, after all the structural stuff, we're going to dive right now into the different groups of plants, and we're going to start with the most basic groups of plants, which we deem as the seedless plants. Now, where do plants come from? Well, we're thinking about land plants here, right? Oceanic plants have existed for quite a bit of time, but land plants are, um, you know, not exactly an old thing, but not exactly a new thing. So they're kind of old, but they're kind of new. Now, how did land plants come to be? Well, all green algae and the land plants actually share a common ancestor that's about 1 billion years old. Now, for a plant to move from the ocean or to move from a lake into onto land requires some really interesting um, evolutionary inventions. Now, the first is water loss. You can imagine if you're living in water, well, you don't have to worry about water loss. But if you're on land, you have to worry about water loss. Now, we talked about the stomata as being a way to prevent water loss, but that was something that had to evolve first. And then you also have the harmful protect. You have to protect yourself against the sun. When you live in the ocean, you don't have to worry about the sun so much. Right? The water protects you from the sun, but when you're on the land, you got to protect yourself from the sun. And the other part of living on land, which I haven't written here, but I think is a really interesting one, is actually gravity. You have to be able to support yourself, right? If you're going to be a 50-foot tall tree, you have to have the structures to support yourself, and those structures have to evolve at some shape or point. So that's what we have here for our overall uh, adaptations. Now, one of the more important adaptations for evolution of life on land um, was actually fungi. Now, fungi actually helped plants colonize land. They did this by helping make nutrients available to plants. So these would be our mycorrhizal fungi that we talked about in the fungal lecture. Again, acting as an accessory root system for these plants, helping them grow and uh, haploid and diploid stages. They trend towards more diploid embryo protection, and they trend towards smaller haploid stages, unlike our algae and like many of the protists that we discussed. So this is a phylogeny of our plants. We have our red algae, our chlorophytes, and our charophytes, and then that leads into all of our land plants. And we're going to talk about all of the 7 groups: the liverworts, the mosses, the hornworts, the lycophytes, the ferns, the gymnosperms, and the angiosperms. And so we're going to talk about the rest of these groups today. Um, but as we mentioned, adapting to terrestrial life involved a number of different things. So protection from drying out or desiccation. We needed to evolve the waxy cuticle and stomata. We also had to move water using tracheids. So tracheophytes, as you'll notice here, so including lycophytes and so on and so forth, have tracheophytes, which are xylem and phloem, using to conduct water and food. You have to deal with UV light, and we have to shift to a dominant diploid generation as well. So those are our adaptations from an evolutionary perspective. Now, As we discussed with protists, plants actually follow that haplodiplonic life cycle. We won't dive too much into it again, but this is the same exact slides basically as I gave you from when we talked about protists and fungi and their haplodiplonic life cycle. But again, it's haplodiplonic is just an alteration between a haploid stage, which we call the gametophyte stage, and a diploid stage, which we call the sporophyte stage. And again, this is a physical representation of what this looks like. So you want to make sure you always keep that in the back of your mind. Now, all land plants are haplodiplonic, as we discuss, and they have relative different sizes of generations, and they vary between them. So mosses have very, very large gametophytes, small dependent sporophytes, whereas angiosperms, being the most advanced plants, have small dependent gametophytes and very, very large sporophyte generations. So let's dive into our first major group of plants on land, which we call the bryophytes. These are the closest living descendants of the first land plants. So they are evolutionarily speaking the oldest. Now, these are non-tracheophytes, i.e. they lack vascular tissue. They do not have xylem and phloem. But they do have other types of cells, which we call conducting cells that help move things around them. Now, the important thing for bryophytes is actually those fungal mycorrhizal associations, allowing them to have water uptake. So in our bryophytes, we have our liverworts, mosses, and hornworts. So let's dive into those. Now, there are some characteristics of our bryophytes. They're very, very simple, but they're adapted to very diverse terrestrial environments. Now, there are about 16,000 species spread across 3 clades, the liverworts, mosses, and hornworts. Um, their gametophyte generation is photosynthetic and conspicuous, and their sporophyte generation is small and dependent. So sporophyte, remember, is the diploid, gametophyte is the haploid. And these groups of organisms require water for sexual reproduction. That's an important thing. They have no vascular tissue and they rely on water for sexual reproduction. So let's dive into our first group, which is the liverworts, or the phylum Hepaticophyta. Now, these, as you can see, have flattened gametophytes with liver-like lobes. So you can see, again, they look like like little sausages. They're liver-like lobes, but I like to think that they look like sausages. And about 80% of these look like mosses. Now they form what we call gametongea, which is actually a familiar structure, right? The fungi, some of our fungi had these, which are umbrella-shaped structures, which you can see here, and they actually undergo asexual reproduction. So that's our liverworts. Not a remarkable group of organisms, but again, certainly present in the environment, an important stepping and they do have multicellular gametongea, again, that umbrella shape, that form. And there are 2 distinct types of gametongea here, the archegonia, which is the female, and the antheridia, which is our males. And they have flagellated sperm that have to swim in water. So let's look at this. We have the gametophyte here, so the haploid version, I'm sorry, the haploid version here, and then we have the sporophyte here, our diploid version. So this is our bryophytes, these are our mosses. You see these all over, if you live in the Boston area, they're all over the Boston area. You can find them, you look at them in depth, they all have the same structure. The gametophyte down here and the sporophyte essentially living together, one not being dependent so much on the other. Now we have this level of haplodiplonic life cycle. We have male gametophytes and female gametophytes. They produce egg and sperm. They meet in water, so the sperm makes it to the eggs via water. So these plants rely solely on water for the distribution of their sperm, which leads to fertilization, giving us a 2N organism, which then produces a zygote, which divides by mitosis, producing a mature sporophyte, which has a sporangium on it, which produces spores, which gives rise to haploid spores, which undergo mitosis, producing germinating spores, which lead to male and female gametophytes, repeating the process over and over again. But again, looking back at this picture here, we have the sporophytes, which we see here, and we have the gametophytes, which we see here. So it's multicellular haploid and diploid organisms living together as part of the same plant. So next up, we have our hornworts, which is the phylum Antherpsophyta. within gametophyte tissue. So not two separate structures that we saw with the mosses. They're embedded within the gametophyte tissue, and their cells, interestingly, have a single large chloroplast. So let's uh let's uh let's look into our next group of plants, which are tracheophytes. Now remember, when something is a tracheophyte, unlike the previous three groups of plants we've looked at, they have that vascular tissue, that xylem and phloem. So the first vascular land plant that we know of from the fossil record is a plant called Cooksonia. It appeared about 420 million years ago in the phylum Rhinophyta. It was only, as you can see, a few centimeters tall, about the size of a pin. It had no roots nor leaves, and it was actually homosporous, producing only one type of spore. So this is the early ancestor of our vascular plants. But tracheophytes, i.e. vascular plants, have 7 extant phyla that are grouped into 3 larger clades. The first is our lycophytes, which are club mosses, our pterophytes, which include ferns, whisk ferns, and horsetails, and then seed plants, which include our angiosperms and our gymnosperms, being this third one, the seeded plants being the most complex of all of them. Now, relative to what we just saw, the gametophyte generation has been reduced in size relative to the sporophyte generation. And there's a similar reduction in multicellular gametangia has occurred as well. So we talked about the vascular tissue, so I'm not gonna dive too deep into this again, but I'm putting this here as a refresher. As we sort of go through it, xylem takes things up, the phloem takes things down. It allows enhanced height of our tracheophytes, allows enhanced structure, and all sorts of other structures, including the development of the cuticle and the stomata. So we have the lycophytes. There are about 1,200 species. They're club mosses. They look a little bit like this. You have the pterophytes, which there are about 11,000 species of ferns. The horsetails, which there are about 15, and then the whisk ferns, which there are about 6 species. So these are our 3 extant seedless but vascular plants. So the most basic of all of our organisms. Now, all these organisms have also evolved stems, roots, and leaves. Now, stems, we know, are the above ground structure, the roots are the anchoring ports, and the leaves are the site of photosynthesis. So, this group, unlike what we saw previously, actually has all 3 of these things. Remember, our mosses don't have roots, right? They have rhizoids, but not roots. They don't have leaves. They have leaf like structures, but not leaves. So, I'm actually going to skip this. You can take a peek at this about the origins of these, but I don't think it's particularly important. So, I'm just going to move on. Now, the other evolutionary aspect of our tracheophytes is the evolution of seeds. So seeds are being awesome, tasty things like peanuts and peas and legumes, but they're highly resistant, containing a large food supply for young plants. And we see them in our lycophytes and pterophytes. I'm sorry, we don't see them in our lycophytes or our pterophytes. They simply don't have seeds, but we do see seeds in our angiosperms and our gymnosperms, which we'll talk about. So this is where we are thus far in the tropics. They do lack seeds, and they do look a little bit like mosses, but their sporophyte generation, unlike mosses, is the dominant version. Now, within this group, we have our next group, I should say, not within this group, but the next group, which is the pterophytes. Now, these are our ferns and all the things that resemble ferns. It's sometimes called the allies of ferns. I just prefer ferns and things that look like ferns. They have a common ancestor that gave rise to 2 clades, the whisk ferns, which we see the whisk ferns here in the ferns, and then we also have another group of ferns and horsetails as well. Now, these groups all still form the Antheridia and the Archegonia, and they do require free water for their flagellated sperm. So they're not super advanced yet, but again, they do have some advantages to them. The big advantages being larger actual leaves, vascular tissues, and so on and so forth. Now, within this group, we have the whisk ferns. These are typically found in the tropics. Now, their sporophyte generation consists of evenly forking green stems, as you can see here, without true leaves or actually roots. So they're kind of a weird group within this, but their gametophytes do develop elements of vascular tissue, but there is actually only one known to do so. Then we have the horsetails. There are 15 living species, and they are all homosporous. I.e., they don't produce male or female or plus or minus. They just produce one type of spore, and they're only found in one single genus, Questium. Now, their sporophyte themselves consists of ribs, joint photosynthetic stems, as you can see here, that arise from branching rhizomes with roots at the nose. And they have silica deposits in their cell, forming what we call scoring rush, as you can see here in white. Then we have the ferns, which is the most speciose group here. They're also the most abundant. They have 11,000 species. And ferns actually gave rise to coal deposits about 300 million years ago that we now use to power homes and all that fun stuff. Now, ferns have a life cycle that we're going to want to discuss, and it differs from that of a moss. So let's dive into it. So we'll start off here where sperm, the antheridium, meets egg, the archegonium, and fertilization occurs, producing a really interesting structure. We have the end tissue surrounding it, right? So the gametophyte around it, and then we have the sporophyte or the egg being 2N. Now the sporophyte, I'm sorry, the gametophyte, yep, no, I was right the first time. The sporophyte undergoes mitosis, producing a larger 2N embryo, which then develops as a leaf of a young sporophyte, which then develops into a mature frond or a as we can see here, this is a frond of a fern, and the adult sporophyte. Now, on the underside of the leaf frond, we have what we call sporous clusters, where we produce the sporangium, which is just one type of spore. So these are homosporous, which then develop into mature sporangiums, which undergo meiosis, producing haploid cells, leading to sort of this overall structure here, which develops into the gametophyte generation. Now, as you can see in the gametophyte generation, there are rhizoids, so those root-like structures. And then on that, we have the antheridium, which is the male parts producing the sperm, and the archegonium, the female parts producing the egg. So it's kind of a weird thing, right? You have this structure all happening on the same organism at the same time, right? This part develops, but again, remember this, the gametophyte generation has the sporophyte generation grow directly out of it. It's kind of a cool thing, right? So it all happens on one organism. There's not distinct plants as well. So I think that's a pretty cool thing. Now, sporophytes, as we saw, have rhizomes. Now, their fronds or their leaves actually develop at the tip of a rhizome as tightly rolled up coils called fiddleheads. So we have the fiddlehead here. This is what a fern looks like. And then it starts to essentially uncoil. I always thought it kind of looked like the octopus, like an octopus arm. It uncoils, forming the mature fronds that we see. Now, as we mentioned, they have a really distinct reproductive system as we've already went through. So that's our ferns, that's our vascular plants, but that are not seeded. But the most important group, arguably, to all of plants is the seeded plants. These are the plants we rely on to build our homes. These are the plants we rely on for sustenance. These are the plants we rely on to feed our livestock, is the seeded plants. Now, seeded plants came to rise on the planet about 320 million years ago, and they did evolve from spore-bearing plants known as the progymnosperms. Now, the success of their evolution was actually contributed to the evolution of the seed. Again, seeds being like beans or peanuts, right? Nutrient-rich things that are tough, but provide the brand new generation, right? If you took a bean that you got from the store and you planted it, it would grow into a full plant, right? That's one of the cool evolutionary aspects of a seed. Really interesting evolutionary adaptation. Now, the seed protects the embryo. Now, the embryo is protected by what we call the integument. Now, we'll just look at the structure here. You can read this on your own. But we have the embryo here in the center. So that's going to be the fertilized egg, essentially. Then we have the integument, which is the outer seed coat. And then we have the endosperm, which is basically the storage area for the seed, right? Stores all the energy. Now, when we think about why seeds are healthy, why beans are healthy, why peanuts are healthy, well, it's actually because they have large endosperm, i.e., lots of nutrients stored in this compacted area. Now, within our seeded plants, there are two distinct types of gametophytes, the males, which we call pollen. They're typically dispersed by wind or a pollinator. There's no need for water here. Everything else we discussed thus far needed water, but we don't need water for our seeded plants. And then we have the female gametophytes. They develop within in a structure we call the ovule, and they're enclosed within a diploid sporophyte tissue in angiosperms. And so within our seeded plants, so our vascular plants that are seeded, there are 5 distinct phyla. the Coniferophyta, the Cicadophyta, the Gnetophyta, the Ginkgophyta, and the Anthophyta. And we're going to talk about all 5 of these because they're pretty cool. Now, we'll talk about the conifers first. Now, there are about 630 species. You guys see conifers all the time, right? If you've seen a pine tree or a spruce tree, that's conifer. Then we have the cycads. There's about 300 species. They're mostly found in tropical regions, but again, they're palm-like. Then we have the gnetophytes, mostly found in dry areas, but there are only 65 species of them. Then we have the ginkgophytes, which are the ginkgo trees, so Ginkgo bilboa. There's only 1 species of ginkgos, just as a note. We actually have them in the Boston area. Ginkgos are pretty cool. They're very pollution tolerant, which is why they're commonly found in cities like Boston. And then the final group, which is the Anthophyta, which there are well over 300,000 species of them. These are the things that you know and love: peanuts, apple trees, tulips, roses, right? That's our Anthophyta, the angiosperms. So we'll dive into all these, but let's start by talking about the gymnosperms. Now, the gymnosperms of this group actually comprise the Coniferophyta, the Cicadophyta, the Gnetophyta, and the Ginkgophyta, and then the angiosperms just encompass the Anthophyta. Now, they're grouped together because they all have seeds, right? These are vascular seeded plants. So these our gymnosperms all lack flowers, which our angiosperms, as we'll learn, have, and they do have an ovule that is exposed on a scale. So you think about pine cones, and we'll get to that in a second. So let's talk about the first group, which are the conifers, which is the phylum Coniferophyta. Now, these are the most familiar ones. These are pine trees, they're spruce trees, fir trees, cedars, and other things like that, including redwoods, which are the tallest living trees on the planet, as well as bristlecone pines, which are the oldest living trees that we know of. Now, they're commonly found in colder environments, but you'll also find them in sometimes drier regions. But again, we typically associate conifers with colder environments such as, Canada and Massachusetts and so on and so forth. Now, these are actually important sources of major products, including timber, paper, resins, and pharmaceuticals such as Taxol, which serves as anti-cancer properties. Now, within the group of conifers, we have the pines. There are about 100 species of pines, all found within the northern hemisphere. And as you guys probably know, they produce needle-like leaves in clusters. Now, these leaves have a very thick cuticle and recessed stomata to help retard water loss. And these also have canals with resin to deter insects as well as fungal predators. Now, pines reproduce with 2 distinct types of structures. The first are male gametophytes, or the pollen. Now, these develop in microspores in male pine cones via meiosis. And then we have the female pine cones, which form the upper branches of the same tree. So they are on the same tree. Males are lower, females are much higher. Now, the female pine cones are the ones that you guys know and love. They make that satisfying sound when you step on them, right? They're larger and they have woody scales, and their 2 ovules develop on each scale. And each contains a megasporangium that is known as the nucellus. Now, let's dive into the female pine cone. Let's dive into the structure. So we have one single sporophyte, so it's a 2N plant. It's developed from a seedling. Now, on the same tree, on the higher branches, we produce the pollen-bearing cones. These are the ones you kind of know and love. And then we produce um, the pollen-bearing cones on the lower ones. These are the ones you don't quite, see so much. They're smaller. They're sort of insignificant in size. They're very, very, very tiny. And then we have the ovulate or the seed-bearing cones, which are the ones that are higher up. Those are the ones you know and love as pine cones. Now, within the pollen-bearing ones, we have the scale, which develops microspore mother cells, which undergo meiosis, producing microspores, which then differentiate into pollen, which have what we call an air bladder, which allows them to be essentially float through the air. Now, within the female cones, we have the megaspore mother cell, which undergoes meiosis, producing the megaspores, which then develop a pollen tube. Now, once so for fertilization to occur, pollen has to make it to the cones via the air. Now, the pollen will then differentiate, producing a pollen tube, which it sends sperm up. But basically, it enters the pollen tube on the female structure, the female cone, leading to it essentially piercing this layer and then producing the pollen tube, allowing it to essentially send pollen up to fertilize the egg. Now, the egg itself develops into a zygote, and it's typically about 15 months after pollination. A section of the seed, which we call the second year, actually shows embryo embedment in the what we call the megagametophytes, which then undergo mitosis to produce seeds, which then those seeds could be planted in the ground, which develop into seedlings, which are just young plants, which repeats this process over and over and over again. So a relatively long reproductive cycle for our pine trees. But yeah, this reproductive cycle is pretty consistent of what we see in our conifers. Um, now this leads us to our next group of our seedless plants, but non-flowering seedless plants, the cycads or the phylum Cycadophyta. Now these are very slow-growing gymnosperms found in tropical and subtropical regions. Now their sporophytes actually resemble palm trees. Palm trees are fun. And the female cones, surprisingly, can weigh upwards of 100 pounds or about 45 kilograms. And they actually have the largest sperm cells of all known. Again, you can see they're found in arid regions. They're really unremarkable plants. Like I feel like if you came across this on an average everyday basis, they would look like it was just a dead plant. But this is just what they look like. They produce cones. They have the same life cycle as our conifers. And then we have our final group, which is the ginkgophytes, Ginkgo bilboa. It has flagellated sperm. It is dioecious, which we have male and female reproductive structures form on different trees. So it's not like our conifers where male is at the bottom and females are on the top. Our ginkgophytes, there are male ginkgo trees and female ginkgo trees. So kind of a fun thing. Now, this leads us into our most exciting group of plants, which we call the angiosperms. And again, these are the plants that you guys know and love, the plants that are tasty to eat, and so on and so forth. Now, the angiosperms are really designated by their capacity to produce flowers, which is a really evolutionary successful thing. Now, the other thing I'll make mention of is their ovules are actually enclosed in diploid tissue at the time of pollination. So we can actually physically look at this. We have the ovules, they're enclosed in diploid tissue, as we see here in this pea uh, pod. There we go. That's what it is. Now, angiosperms are really remarkable. They are, as you saw, the most speciose group on the planet, but they're also the most abundant group of plants on the planet. And they really, with their arrival, it really dramatically changed the planet. Really did. When angiosperms popped up in the fossil record, we actually saw a massive explosion in the evolution of other species because once flowers were a thing, pollination also of those flowers also needed to be a thing, which led to the evolution of numerous species of insects and birds and so on and so forth across the planet. So angiosperms are really important for the evolutionary kick that gives us our really diverse group of organisms we see on the planet today. Now, how angiosperms came to be, your guess is as good as mine, but the earliest known angiosperm is actually in the fossil record from 125 million years ago in a group called genus called Archaeofructus. We don't think this was the first angiosperm because it does lack sepals and petals, as we'll discuss in a moment, but this is the first one we see in the record. So 125 million years is the bare minimum in terms of how long it's been, but they're likely probably older. Some people say 200 million years, other people say 150 million years. We really don't know at this point in time. Now, we just finished talking about the gymnosperms. Now, within the angiosperms, there are numerous groups, as you'll see. Now, the Archaeofructus, which is the extinct version, there are 6 groups we're going to talk about. The monocots, which we've sort of been alluding to, the magnolids, so magnolias, the eudicots, which again we've been talking about. We have the star anise, the water lilies, and the ambarella. And we're going to talk about all these. But we'll first talk about the closest relative to the ancestral angiosperm, which is the ambarella. Now, we can see an ambarella here. It has carpel, it has petals, it has sepals. We'll get into the structures here, but this is our oldest relative of all angiosperms. And there seems to be a really interesting evolutionary thing where mitochondrial genes were transferred from mosses to flowering plants. Actually, a pretty cool evolutionary thing. Not so important, but I wanted to just make mention of it because I think it's a pretty cool thing. Now, as I mentioned, The key innovation here for angiosperms is flowers. And flowers house the gametophyte generation. So the rest of the plant is diploid, but the gametophyte being haploid is housed in flowers. So let's look at these flowers. And I'm going to skip this like text slide. I'm just going to physically show you the flower. And I think it's much, much easier to just not see this stuff and physically see this. Now, excuse me, we have the stamen, which is comprised of the anther, which is the outer part, and the filament. Now, this is going to be the male parts, i.e., where the pollen is produced. Then we have the carpel, which is comprised of the stigma, which is the outer part here, the style, which is a tube that leads down to the ovary. Now, the ovary is comprised of the ovule and the ovary wall. And then we have this part that's called the receptacle, which attaches the ovary to the pedicel, which, as we will recall, the pedicel attaches to the stem, right? So we have on a single flower, and you've seen this before, maybe you didn't realize it, but on a flower we have pollen and we have ovaries on the same flower, the male and female structure. Now, the only other two structures I want to make mention of here, actually the only two structures left on this diagram, is the sepal, which are essentially the green leaves that surround a flower. So if you've ever picked up a flower, you'll notice there are green leaves on the flower. Those are what we call the sepals. And then we have the flower petals, which are usually brightly colored, designed to attract pollinators to them. So this is the structure of our plant. This is the structure of a flower. Know this. This is really important. Now, there's a really interesting part here, which we call double fertilization. And I'm actually going to skip over these slides to actually just show you it. I think like it's easier to understand by just using a diagram to understand what's going on. So we start off here with a megaspore, and we have megaspore being the female parts, and then we have the male parts here, right? So the anther. Now, both undergo mitosis. The first one produces pollen. That's what the sperm is, right? And then the megaspore, which is the female parts, undergoes mitosis, producing the egg, sort of the way we think about it as an egg. And then we have what we call the polar nuclei. Now, fertilization occurs by pollen making its way into the style down there, producing essentially like a sperm shape where we have a pollen tube attached to the sperm. And that basically allows the pollen to basically eject the sperm into the egg. So that's what's going on there in terms of fertilization, which then leads to the sperm again coming there, and the sperm does two things. It first fertilizes the polar nuclei and the egg at the same time. So this is why we call it double fertilization, because we're fertilizing not just the egg but also the polar nuclei. Now, that double fertilization event takes us from our haploid stage over here, leading us to our diploid stage where we have the zygote, which will develop, as we've seen before, into the embryo, and then the polar nuclei, which will then develop into the endosperm, that really sort of tasty, nutrient-rich part of the seed. Now, once the seed is produced, it can germinate, i.e., you toss into soil and it starts to germinate into a young sporophyte, which then develops into a mature sporophyte, which then produces flowers, repeating the cycle over and over again. So our double fertilization event, which is again unique to our angiosperms, occurs to produce the two distinct structures of a seed that we've mentioned, the embryo and the endosperm. The embryo developing into the plant, the endosperm being the very nutrient-rich part of a seed. So it's kind of a cool thing, right? Um, Now, pollen production is much simpler. So sperm, as across all organisms, is really simple to produce. It's similar, but significantly less complex than the female gametophyte production. Um, and Pollination as a process is, again, the mechanical transfer of pollen from the anther to the stigma, which may or may not be followed by fertilization. And as I mentioned, pollen grains develop into a pollen tube that is guided to the embryonic sac. And then one of the two pollen grains lags behind, um, again, lags behind, and this cell actually produces two sperm cells, which have no flagella on them. And again, double fertilization, as we discussed, is really important for the production of angiosperm seeds. Again, this is just text version of what we saw, just as a note. Now, seeds are really important, right? We eat a lot of seeds as part of our diet, but seeds help help plants propagate themselves, right? Essentially a seed can be dormant for an extended period of time, which is kind of a fun thing. A seed is tasty, it's packed with nutrients, and plants use a seed as a mechanism to spread their seeds away from their body. Now, we can actually physically look at the structure of a seed developing. So we have the overall seed coat here on the outside, which is also called the integuments. Now we have the overall structure, right? We have the embryo here in blue. Now there are a few structures to the embryo here. The first is the root apical meristem, which we've already discussed, and the root cap. Then we have the procambrium, which again we've already discussed. And then we have the shoot apical meristem. And then the other part of the plant, which you've seen on figures in this talk before, but we haven't quite mentioned until now, which are called the cotyledons. The cotyledons help act to form sort of the first leaves of these plants once they've developed. And then we have the endosperm, which again is that really nutrient rich that fuels the growth of this young plant once it's once it's planted in the ground. Now, seeds protect embryos by maintaining dormancy under unfavorable conditions. What is dormancy? It's just when something is asleep, right? That's all dormancy really is. They also protect the young plant when it's most vulnerable. Really important thing. Protection is important, as we saw with the meristems. Things that are young and developing need protection. They also provide food for the embryo until it can photosynthesize, and they also help facilitate the dispersal of the embryo. They also allow the seeds to pass through the digestive system of organisms, right? So you can eat a seed and poop it out. Why is that? Because the seed coat protects them. Now, the seed coat, once it forms, most of the embryonic metabolic activity cease to exist, and germination of the seed actually can't take place until oxygen and water reach the embryo. So some seeds of some plants actually have been known to remain viable for thousands of years, which is kind of a wild thing. As long as they don't get wet, they basically stay viable and dormant, which is really wild, right? Just picture finding 1,000-year-old seed, planting it, and it grows, right? That's the sort of level of protection the seed coat provides. Now, once a seed lands in the ground, it will germinate into a young plant, which then develops into an adult plant, right? But germination is a really interesting thing. There are specific adaptations that ensure seeds will actually only germinate under appropriate conditions. Now, for instance, some seeds actually lie dormant within tough cones that actually will not open until they're exposed to fire. So many of the conifers that live on the California coast are actually have evolved this capacity that will only germinate seeds once a fire has come through the area. So germination is kind of a cool thing. This is one specific adaptation of seeds. There are many, many more out there that you can certainly explore for yourself. But I just want to make mention of the fire ones because it's always a relevant thing because California and the California coast always seems to be on fire because of climate change and all that fun stuff. So now with our angiosperms actually came the evolution of one of the best things about plants, which are fruits. Now, fruits are simply defined as mature ovaries or carpels. So when you eat an apple, all you're eating is mature ovary. That's it. When you eat a mango, mature ovary. But within that fruit, you have seeds. Now, the way fruits form is that during seed formation, the flower ovary begins to develop into a fruit, which is kind of a weird thing. Now, it is possible for fruits to develop without seed development. For instance, bananas are propagated asexually, so they have fruits, but they don't have seed development. It's kind of an interesting thing. But let's look at how a seed develops um, um, into this. So first off, a fruit development, not a seed development. The ovary wall is termed the periocarp, which has three layers: the exocarp, the mesocarp, the endocarp, and their fate determines the fruit type. Now, fruits contain three genotypes in a single package. The first is, which is kind of really interesting. Now, the fruit in the seed coat form prior to the sporophyte generation. The remnants of the gametophyte generation produce the egg, and the embryo represents the next sporophyte generation. So within a single thing, we have three distinct generations, three distinct genotypes. One is a hybrid, one is the old, one is the new. Kind of a weird thing to think about as a structure. So that's what we have here. Now, within our fruits, there are many distinct types of fruits that produce. The first category is what we call the true berries. Now, in this case, the entire periocarp, as we can see here, is very thin skin. It's fleshy. They have multiple seeds in one or more ovaries. And for instance, the tomato plant is actually has fused carpels that are then attached to the seed. So tomatoes are berries, whether or not we typically don't think of tomatoes as being fruits, but biologically speaking, they are true berries, thus they are fruits, not vegetables. Then we have legumes. Now, legumes, where we have the fruit physically splits along the carpal edges, as we see here, giving us two distinct sort of structures. We know these best for beans and peas. Now, unlike our fleshy fruits that we just saw, their three tissue layers of the ovary wall actually don't thicken extensively, and the entire pericarp is dry at maturity. So, our legumes dry as they age. Then we have drupes and samarans, drupes being things like peaches and plums and cherries. Now, in this case, we have the seed, which has a very, very a hard outer area called the endocarp, which is called the pit. So I'm sure you guys probably had a peach at some point in your life, or a plum. You know, they have the pit. That's the endocarp. Very, very, very tough. Like you can run over your peach pit with a car and it doesn't break. That's how tough it is. But then they has the mesocarp, which is the fleshy part, and the exocarp, which is the sort of the skin. And as you guys probably know, the exocarp on peaches can have fuzz to it. Then we have samaras, which actually the fruit doesn't actually split at all, and a wing forms, which we call the pericarp, which is part of the pericarp, allows it to spin. And we see this in maples, elms, and ash trees as well. So you've seen these before around the Boston area. They are fruits, which we call samaras. Excuse me. And then we give to our final two fruits, which we have the aggregate fruits, which include strawberries, blackberries, raspberries. These are not berries. Tomatoes are berries. Strawberries, blackberries, and raspberries are not berries. They are aggregate fruits. Now, aggregate fruits are derived from many ovaries of a single flower, and the ovaries themselves are not fused. So we have the seeds here, we have the ovaries, but they're distinct ovaries on one plant, which is why they're aggregate fruits. I hope that didn't make you sad that your blackberries are not actually berries, but tomatoes are berries. I know it's kind of a weird thing, but that's just the way they are. And then we have our final one, which is our multiple fruits. And the best case for this is pineapples. They have individual flowers that form around a single stem. So we have the main stem here, and we have our individual flowers here. Now, if you guys have never seen a flowering pineapple, I suggest you Google it. But a flower will pop up on each one of these little spots here, which then leads to all the reproductive parts of an angiosperm that we've discussed. So that's our multiple fruits, classically highlighted by our pineapples. Now, why do plants produce fruits? Well, fruits are awesome. They're not just awesome for us, they are awesome for other things to eat. So fruits have evolved as a thing to disperse seeds. Seed coats are tough, but fruits are easy to eat. So by eating fruits, animals ingest seeds, thus taking the seeds away from the angiosperm and pooping them out somewhere else, allowing the plant to disperse itself over a geographic area. So that's how we have fruits. They're not just tasty things, they are ways to disperse your seeds. Now, the other way other ways I should say, we commonly see seeds being taken away, i.e. fruits being taken away, is the wind. As we saw with these samaras, right? They're shaped like wings. It helps them to spin through the air and be carried by the wind away. Now, we also know that others can actually be hitch a ride with hook spines. And we can see that here in this plant here. It has these little hooks that attach onto fur or even the clothing of humans, and you take it away from you and it falls off eventually, dispersing the seed away from the adult plant. And we can see coffee here. Again, tasty thing to eat. The seed is very tough. An animal eats it, takes the coffee seed away, poops it out, dispersing the seed away. So that's the purpose of our fruits. Tasty things to carry things away in some shape or form away from the maternal plant. Now, this leads us to not talking more about groups, but actually how plants move materials. So this will be the final part of our plant lecture today. I know we've gone through a lot today, but we have more to go through because plants are really, really complex. So we're going to ask the question now, how do plants move water up the top of a 10-story high tree, right? I have oak trees in my backyard right now, and they are about 75 feet tall. How does a plant move water from the roots up to the shoots? Well, first thing that has to happen, the water enters the roots. Then it moves into the xylem, which is, as we know, is the innermost vascular tissue. And then the water rises through the xylem because of a combination of factors. And then most of that water ends up exiting through the stomata in the leaves. Now, long distance movement in plants um, is a result of essentially long distance movement of materials. Now, water and dissolved minerals travel great distances in the xylem and the phloem, but we're thinking about going up because going down just has gravity, right? Now, for things to get up, they essentially get pulled by transpiration. So as leaves get baked by the sun, the water inside them heats up and eventually evaporates. This process of evaporation of water inside of a plant leaf is called transpiration. So as the water in the plant leaves evaporate, it transpires out of the plant leaf, leaving through the stomata. And this causes a pulling force, allowing water to be pulled up through the xylem from very, very deep distances all the way up to the top. So that's one way. Transpiration pulls things. Now, the other way is actually cohesion and adhesion of water. So water molecules stick together and then they adhere to walls. And they kind of do this back and forth dance where they stick to one another, attach to the walls, pulling themselves up with the aid of the forces of transpiration. So kind of a cool thing. And we can physically see that here, structurally, water comes in, it goes into the xylem, it gets pulled up by transpiration and evaporation, as well as the cohesive and adhesive forces of water, pulling it up, eventually exiting as either, well, I should say, either being used for something or exiting via water vapor through the stomata. So kind of a cool thing. Now, movement of water at the cellular level basically tells us that water is moves relatively easy. As you probably know, water diffuses through plasma membranes by osmosis, which can be enhanced by proteins called aquaporins. Now, other substances also depend on transporters, so movement of other things in the vascular system. Now, as you probably know, things can be moved from inside to outside of a cell or vice versa via facilitated diffusion, or what we call active transport. Can also be moved by ATP powered hydrogen pumps or unequal concentrations used to drive osmosis. And this is how we transport things between cells. These were, we went over these in depth quite a bit in Bio 1, or maybe not with me, maybe with another teacher. But this is how we move things across membranes and plants. In particular, movement of water is actually heavily tied to this concept of water potential. Now, water potential is a way to represent free energy. So water potential, which we define as this Greek character that always escapes my name of what to call it, is used to predict which way water will in fact move. Now, water moves from areas of high water potential to areas of low water potential, and it's measured in units of megapascals. And so that's water potential. Now, water commonly moves through these proteins that are embedded in cellular membranes called aquaporins. Again, just a channel to move waters. It's kind of a pretty cool. It's selective. It only allows water to move across. And again, it allows the facilitated diffusion of water. Now, um, just as a note, there's a lot more about water potential that we're not going to cover. It's a lot of physics and stuff like that. But just to know, that's the way water moves via water potential. Now we have transpiration, i.e. the evaporation of water. And the really kind of wild thing is that over 90% of the water taken in by a plant's root is ultimately lost to the atmosphere. So evaporation takes it away. So it's kind of a weird thing, right? Imagine if 90% of the water you drank, you sweat it out, right? That's not the case, but imagine if that was the case. That's what's happening here. Now, at the same time, photosynthesis does require CO2 and supply as a CO2 as a supply from the atmosphere. Now, as we discussed, the stomata regulate water loss. So they open and close. So when they're open, water can go through. When they're closed, water cannot go through. But remember, a plant needs to have those stomata open to uptake CO2 as well as expel oxygen. So the stomata has this tough job. It has to be open, but it can't be too open because too open leads to too much loss of water. Now, so as we discussed the stomata, they have these guard cells, which they're epidermal cells containing chloroplasts. They have very, very thick cell walls on the inside and thinner cell walls elsewhere. And what these guard cells do is they bulge and bow outward when they become turgid, which causes the stomata between two guard cells to open. Now, the turgor, or i.e. just a pressure in the guard cells, actually results from the active uptake of potassium and chloride and malate, which is a type of carbohydrate. What this does is adding these solute causes the water potential to drop, thus ensuring water enters osmotically and the guard cells become turgid, i.e. opening them up. Now, what does that look like? Well, in this case, the guard cells are not turgid. They're kind of flaccid, essentially, as you can see in this balloon example here. But to what ends up happening is that um, these stomata cells, these guard cells, when they want to open, they flood ions into these guard cells, allowing water to go in, essentially inflating these cells, very much. That's a stomata. Now, you might ask yourself, what affects stomatal opening besides um, the desire to exchange gases? Well, stomatas open and close when close when CO2 concentrations are very high. They also open when blue wavelengths of light promote the uptake of potassium by the guard cells, and they actually close completely when temperature exceeds 34 degrees C and water relations are unfavorable. Now, you might imagine, well, lots of stuff on this planet lives above 34 degrees C, right? The desert is consistently above 34 degrees C. So how do plants in the desert have stomata? Well, there's actually some alternative photosynthetic pathways, such as what we call crucillian acid metabolism, or CAM, that help reduce transpiration. We'll talk about those soon. Now, in addition, water stress is a big deal for plants. So there are many morphological adaptations that allow plants to limit water loss in drought conditions as well, including going completely dormant, losing their leaves. We see this in deciduous plants, covering their leaves with a much thicker waxy cuticle and woolly trichomes, reducing the number of stomata, and having stoma in pits on the leaf surface, trying to reduce their water stress. And we can physically see this here. This is a plant that's suffering from drought, but it's producing trichomes, producing essentially sunken in stomata, again, to try to help reduce water loss. So that's our protections from drought. Now, flooding is equally as dangerous to plants. Now, plants need oxygen, and they need oxygen in their roots. Now, when water floods soil, it causes soil to go completely anoxic, i.e., no oxygen. So plants have evolved many adaptations to allow them to survive flooding. Now, we commonly see when plants are exposed to flooding some really abnormal growth. We also see oxygen deprivation being a very serious problem, as well as um, plants that live in fresh water have actually evolved some interesting structures in called a new type of tissue, which we call uh, erenchyma, which is loose parenchymal cells with large air spaces, which allow them to collect oxygen and transport into submerged parts of the plant. And we can see this in our lilies here. We can see our site, our stoma here, our upper epidermal. We have the veins. And then again, we can see these erenchyma with lots and lots of air spaces, allowing them to capture oxygen, transport it through their veins down to the vascular system, down to the lower parts of the plant, which are anchored in anoxic sediments. Now, saltwater is another difficult thing to handle. Plants such as mangroves or salt marsh plants, they grow in areas that are flooded with salt water. Now, being flooded all the time is a really difficult thing to deal with, so they have to supply oxygen to their submerged roots. So we talked about nematophores in mangroves before, long, spongy, air-filled roots that emerge above the ground. Now, they also have evolved succulent leaves that contain large amounts of water to help dilute salts, and plants have also evolved the capacity to secrete salt or block salt uptake as well. And again, we can see our mangroves here. It's normal garden variety tree, anoxic sediment, producing these pneumatophores that come up, allowing them to take oxygen out of the air and pump it down to the rest of their extensive root system under here. And you can see for the single tree how big its root system is. It is quite big, which is pretty cool. Now, growing which is another interesting aspect of evolutionary of our plants. Now, this is where most carbohydrates produced in plant leaves are distributed throughout the phloem to the rest of the plant. Now, this process, which we call translocation, provides the building blocks for actively growing regions of the plant. Now, just to sort of show you what this looks like, aphids, which are just insects that suck phloem up, they basically pierce the phloem, which again is taking nutrient-rich stuff from the roots. I'm sorry, the shoots down to the roots. The aphids have evolved to take advantage of that and suck up this nutrients. Again, showing how important phloem transportation is. But phloem transports a lot of other stuff than sugars, including hormones, RNA, sugars, amino acids, proteins, and so on and so forth. So with that, I'm going to end this part of the lecture because at this point, this lecture's gone 190 slides and quite a long distance. So I'm going to end today's lecture here. We'll pick up on plants part 2, covering the remaining part, just talking about soil and so on and so forth. I hope you learned a little bit today. I hope you guys have a great day. Take care.